Hello, my name is Rathmann and I'm a bioarchaeologist working at the University of Tübingen. In the next couple of minutes, I would like to explain how we can use the shape of human teeth as a proxy for DNA in order to reconstruct genetic relationships between archaeological human remains. The shape of human teeth is heritable, highly diverse, and varies among individuals and populations across the globe. Moreover, teeth are the hardest tissue in the human body, and dental remains are generally well preserved in archaeological contexts, even when associated skeletal and DNA preservation is relatively poor. As a result, dental remains are widely used for inferring the biogeographic origin of a deceased individual and for reconstructing ancient human population history, especially when no other biological data are available. On this slide, you can see some examples of dental morphological traits that are commonly used for reconstructing genetic relationships. Examples of dental traits include the number of cusps and roots, the relative size of cusps, or the pattern of ridges and grooves on tooth crowns. Dental traits like these are highly heritable and they occur at different frequencies across populations. These traits can be collected with the Arizona State University Dental Anthropology System, or in short, ASU-DIS, which allows for a standardized scoring and data collection procedure. The system currently comprises a set of 42 dental traits that can be observed in the permanent adult dentition. However, there's disagreement as to how these dental traits evolve and whether certain dental traits are more useful than others for reconstructing genetic relationships. Most experts assume that human dental traits are selectively neutral and that the worldwide human dental diversity was generated by random evolutionary processes consisting of founder effects and genetic drift. By contrast, it has also been proposed that some dental traits evolved in a non-neutral manner, as a result of natural selection and adaptation, perhaps in response of chewing behavior or environmental factors. Importantly, reconstructions of human population history should focus on neutrally varying traits because local selection can create confounding population structures that are unrelated to genetic drift. Until now, it is unclear which dental traits or trait combinations are the most useful for capturing neutral genomic variation. As a rule of thumb, it is therefore assumed that phenotypic analysis based on many dental traits are more reliable than those based on only a few traits. This assumption, however, has never been formally tested. My colleague Hugo Reis-Centeno and I addressed this research gap, and we systematically tested the utility of severally common used ASU-DIS dental traits and millions of dental trait combinations in reflecting neutral genomic variation in a worldwide sample of modern human populations. We published our results in the journal PNAS in May 2020, and in the next couple of slides I would like to highlight our research strategy and main findings. You can find a link to the article in the descriptions below if you're interested in further details that are not covered in this video. A promising approach to test the utility of a specific morphological trait or trait combination is to quantify the correlation of biological affinity measures across worldwide modern human populations, derived independently from neutral genomic markers on the one hand and morphological traits or trait combinations on the other hand. Our analysis was based on a large microsatellite loci database comprising information about 645 mean allele sizes and a large ASU-DIS dental trait database comprising information about 27 dental trait frequencies. We matched these two databases so that we had matched population samples for 20 human populations that were equally distributed across the globe. To assess the utility of a given dental trait or trait combination, we first estimated a dental phenotypic distance matrix called DP between the 20 worldwide modern human populations. We then estimated a neutral genomic distance matrix called DG among the same or closely matched populations. The congruence between DP and DG was then quantified by linear regression of the off-diagonal values in the two distance matrices using Pearson's product moment correlation coefficient R. Conceptually, an R value close to 1 indicates that a trait or trait combination reliably reflects neutral genomic patterns of variation, whereas an R value close to 0 indicates that a trait or trait combination is less congruent with neutral expectations. To account for stochastic variation inherent to a neutral model of evolution, we calculated R for a given dental trait or trait combination 1,000 times, each time comparing the DP matrix to different DG matrices randomized by subsampling genomic loci. The result was a distribution of R values, and we reported the median of the distribution as a point estimate and as the utility estimator for a given trait or trait combination. To measure the spread of R values around the utility estimate, we constructed an interpercentile range accounting for 95% of the distribution of R values. We also calculated a p-value for the utility estimate. For this, we first estimated a null distribution of R values by comparing 1,000 permuted DP matrices, where rows and columns were randomly rearranged, to loci resampled DG matrices. We then calculated the p-value as the proportion of R values from the null distribution that are equally high or higher than the utility estimator obtained from the observed data. We have then performed this analysis for all 27 dental traits included in our dental database, as well as for more than 130 million possible combinations of these traits. The result was a comprehensive table listing utility estimates, 95% R ranges, and p-values for single dental traits and all possible dental trait combinations. This reference table can be used for future investigations, for example, to validate the performance of a particular dental trait combination observed on a prehistoric skeleton, or, for example, to validate the performance of a particular dental trait combination employed in previous studies. You can find a link to the table in the descriptions below. So let's look at the results for the individual dental traits and how useful they are for reconstructing human neutral genetic variation. Here you can see a bar chart visualizing the utility estimates for the 27 dental traits in descending order. The dots display the utility estimates, bars indicate 95% of the distribution of R values around the utility estimate, and grayscale of dots indicate p-values for the utility estimates. Dental traits shown at the top of the plot are more useful for inferring neutral genetic variation than the dental traits shown at the bottom of the plot. This result is consistent with previous studies suggesting that several dental traits shown at the bottom of the plot evolve in a non-neutral manner as a result of natural selection and adaptation, perhaps in response to chewing behavior or environmental factors. Researchers interested in reconstructing neutral genetic variation from dental morphology should therefore concentrate on traits closer to the top of the plot. Nevertheless, none of the utility estimates are statistically significant at the conventional alpha level of 0.05, indicating that human neutral genetic variation cannot be reliably reconstructed from single dental traits. We therefore wanted to explore whether morphological inferences about neutral genetic variation based on many dental traits are more useful than those based on only a few traits. Here, we plotted the distribution of utility estimates and associated p-values resulting from trait batteries of different sizes using violin plots. So, from single traits to your left to the full 27 trait battery to your right. When you look at figure A, you see that, on average, increasing the number of traits leads to a logarithmic increase in utility estimates, with a gradual tipping point at which adding more traits only adds little new neutral genetic variation. At the same time, increasing the number of traits reduces the variance of utility estimates. Nevertheless, the highest utility estimates were achieved by using a rather limited number of traits ranging from 14 to 20 traits as indicated by the red box, and not by using the full 27 trait battery. In total, we found a set of 267 combinations that all performed equally well in capturing neutral genomic variation best. We propose that these trait combinations should be prioritized in future research, as they allow for more accurate inferences about population history when using dental morphology as a proxy for DNA. You can find a link to the table with the 267 best performing trait combinations in the descriptions below. What is also interesting, when you look at figure B, you see that, on average, increasing the number of traits leads to a logarithmic decrease in p-values. In trait combinations comprising more than 15 traits, no matter which traits of the 27 traits are chosen, are always significant at the conventional alpha level of 0.05. We anticipate that our study will serve as an important reference for a wide range of future dental morphological investigations using traits in the ASU-DIS. And our results allow for more accurate inferences about modern human population history at a global scale. And for this, we advise relying on larger batteries of dental traits and not on single trait combinations. And we advise relying on trait combinations with high utility estimates, or alternatively, to remove traits with low utility estimates. Thanks for watching.